Welcome back, friends. Today we will start a new chapters called proteins. In this lecture, we will discuss about introduction of proteins, also structure, properties, and types of amino acids. So let's start today's lecture. First of all, a bit of introduction about proteins. Proteins are a diverse group of organic compounds, and they are the most abundant biological molecules which occurs in cells and the parts of cells. The term proteins was coined by a Dutch chemist known as Mulder, and it is derived from the Greek words proteios, which means of the first rank. So it is very important. So it is also called as proteios, which is of the first rank. Emil Fischer in 1902 said that all proteins consist of chains of smaller units, and he named it amino acids. So he said that proteins consist of smaller chains of amino acids. Proteins are also a polymer like carbohydrates and nucleic acid, and it consists of smaller unit known as amino acid as monomers. So polymer consists of monomers. Protein is a polymer which consists of amino acid as a monomer. There are about 300 amino acids which exist in nature, but only 20 amino acids are standard and present in each protein, which is coded by the genes. So about 300 amino acids which exist in nature, only 20 are present inside the proteins. The size, shape, and function of proteins are determined by the number, types, and distribution of amino acid and molecule. So the shape, size, and functions of proteins are highly dependable on the number, types, and distribution of amino acids. Next is the structure of amino acids. Amino acids are biologically important organic compounds, and they are composed of amine and carboxylic acid groups along with a side chain which is denoted by R, and this is specific to each amino acid. The side chain is specific for each amino acid. So amino acid consists of amino group, carboxylic group, and the R chain. This is the side chains. The key elements of an amino acid is carbon, hydrogen, oxygen, and nitrogen. The side chain, which is represented by R, can be simple as hydrogen or methyl group. Hydrogen is present in glycine, or it may be a complex structure present in a specific amino acids. The first carbon is a part of the carboxyl group, and amino group is attached to second carbon, which is also known as alpha carbon. The alpha carbon in all amino acid is asymmetric, except in glycine, which is symmetric. So this is the structure of amino acid. You can see here, this is carbon 1, this is carbon 2. So carbon 1 contains the carboxylic group. This is the carboxylic group. Carbon 2 contains amino group, NH2. And this carbon is also known as alpha carbon because it contains 4 different groups. This is hydrogen, this is R group side chain, this is amino group, this is carboxylic group. So it is known as alpha carbon. And the alpha carbon is asymmetric in all amino acid except in glycine, which is asymmetric amino acid. Due to the asymmetric nature of amino acid can exist in 2 optically active form. First one is those amino acids which having NH2 or amino group to the right side are designated as D form, and those amino acids which have NH2 group present to the left side is known as the L form. The amino acid residue in a protein molecule or L form or L stereoisomer. The D form amino acid occurs in small peptides, for example, in bacterial cell wall and certain peptide antibiotics. So the amino acid present in proteins are L amino acid. So this structure you can see here, this it show D and L amino acid. So we discussed that in D amino acid the NH2 group is present right side. This is the right side of the amino acid, and here in L form amino acid is present on the left side. So these are two stereoisomer of amino acids. The first amino acid was discovered is known as asparagine, which is discovered in 1806. The last amino acid which was discovered was threonine. It is discovered in 1938. All amino acid has trivial or common names. In some instances, it is derived from the sources where they were first isolated. For example, asparagine, it is found in asparagus. Glutamate is present in wheat gluten. Tyrosine, which was first isolated from cheese because it is from originated from Greek word tyros. Tyros means cheese. Glycine is originated from Greek word glykos, which is sweet, is named glycine because of its sweet nature. Next is the peptide bond. We know that amino acid combine with each other through peptide bond. Amino acid are held together in a protein by covalent peptide bond or peptide linkages. When an amino group of amino acid combine with the carboxylic group of another amino acid, a peptide bond is formed. Dipeptide has two amino acid and one peptide bond. Tripeptide has three amino acid and two peptide bonds, whereas those peptides which have more than 10 amino acid are known as polypeptides. So this is the typical reaction of formation of peptide bond. This is amino acid 1. This is amino acid 2. So these two combine, water is released. So this is one amino acid, this is another amino acid. This is a dipeptide. It contains two amino acid and one peptide bond. This is a peptide bond which is formed between the carboxylic group of one amino acid and the amino group of another amino acid. And when tripeptides are formed, it contains two peptide bond and three amino acid and so on. Proteins and polypeptides are used interchangeably. So polypeptides are generally have smaller molecular weights which are below than 10,000 Dalton, and proteins have higher molecular weights than 10,000 Dalton. So this is the difference between proteins and polypeptides. Next is there are two important types of amino acid, which is essential amino acid. This type of amino acid cannot be synthesized inside the body. Therefore, it must be present in the diet we consume in order for the protein synthesis to occur. So it is very important that we consume the diet which contain essential amino acid because the body cannot synthesize it. These are 9 in number: 1, 2, 3, 4, 5, 6, 7, 8, 9. So these are essential amino acids. Next is the non-essential amino acid. These amino acids can be synthesized inside the body and that are not necessarily need to be acquired through diet. So the body can synthesize it. These are 11 in number and these are the names are given here: adenine, glutamine, etc. So these are different types of amino acids. Next is the properties of amino acids. So let's talk about some properties. Amino acids are colorless and crystalline solid in dry form. So they are crystalline solid when dry. All amino acids have a high melting point. This is another important property. They are soluble in water and insoluble in organic solvents. On heating to high temperature, they decompose, which is also known as denature. The denaturation of proteins occur at high temperatures. All amino acids except glycine are optically active. Only glycine, you have to remember this, they are optically inactive. Other than glycine, all amino acids are optically active. Amino acid may be sweet, for example, glycine, alanine, and valine. These are sweet. Tasteless, for example, leucine are bitter, for example, arginine and isoleucine. Amino acid contain acidic and basic groups. Acidic is carboxylic group and basic is amino group and can donate or accept a proton, hence known as Impolites. So Impolites are those substances or compounds which contain both acidic and basic groups. Also, amino acid exist as a dipolar ion. Carboxylic group exists as an anion and it have negative charge, and amino group exists as a cation and it has a positive charge, and these two are known as Zwitter ion. Zwitter derived from a German word which means hybrid because these contain cation and anion exist as a hybrid as Zwitter ions. Each amino acid has a specific pH to carry both negative and positive charges and exist as Zwitterian. So you can see here in this figure the ionic form of amino acid. This is neutral or Zwitterian. You can see here this is neutral, negative and positive charge. Also it exists as a cationic form, positive charge it contains, or anionic form contain negative form charged carboxylic group. So amino acid exist as Zwitter ions. Also that pH isoelectric point is a pH point at which amino acid exist as Zwitterian or dipolar ions and carry no net charge. It is definite for every amino acid, the isoelectric point. Here some examples are given. This is the isoelectric point. This is my aspartic acid have 3.0, glycine 6.1, histidine 7.6, arginine 10.8. So it is definite or specific for each amino acid. And this is known as isoelectric point. It is a pH where amino acid exists as Zwitterian ion. Amino acid serves as a buffer and tend to prevent pH change when an acid or base is added because it acts as an acid and base, so it acts as a buffer and prevent a pH change. Amino acid has acidic and basic group and they behave as salts like salts because amino acid has both acidic and basic properties, so it also behaves like salts. The next important topic is reaction of amino acids. The reactions of amino acids are due to the presence of carboxylic and amino group. First one is reaction due to carboxylic group. And this amino acid forms salts. You can see here with bases. So when amino acid combine with bases, it forms salt. And when it combine with alcohols, it forms ester. So amino acid form salts and ester when combined with bases and alcohols. Also, amino acid undergo decarboxylation to produce the corresponding amines. So you can see here, this is an amino acid. So decarboxylation occur, carbon dioxide is released, and you can see here this is amine is produced. Next, carboxylic group of dicarboxylic amino acid react with NH3 to form amide. So you can see here, this is aspartic acid. It contains dicarboxylic group. So carboxylic group of aspartic acid combine with NH3 and form amide asparagine. Next, glutamic acid also combine it and it form amide glutamine. Next is the reaction due to the amino group. Amino group behave as a basis and combine with acid to form salts. So we discussed that amino group behave as bases and carboxylic group behave as acid. So amino group combine with acid and form salts. Next is amino acid react with ninhydrin to form a purple, blue, or pink color complex. This reaction of amino acid with ninhydrin is used for quantitative determination of amino acid and proteins. So it is an important reaction. Next is transfer of amino group from amino acid to a keto acid to form a new amino acid. This reaction is known as transamination, and this is an important reaction in amino acid metabolism. So this is example is given. This is a alpha ketoglutarate, you can see here, and this is an amino acid. So transamination occur, this amino group transfer into this keto acid. So you can see here, and this form glutamate. This is a new amino acid, and a keto acid is formed. So this is the reaction known as transamination. The amino group transfer from this amino acid into this keto acid and form a new amino acid glutamate. Also, amino acid undergo oxidative deamination to release free ammonia. So this is amino acid, particularly this is threonine, and deamination of threonine occur and it form a keto acid and free ammonia. So this is oxidative deamination of amino acid. The next topic is classification of amino acids. Amino acid is classified based on their structure and the structure of their side chain, which is denoted by R. So two basic categories of amino acids are non-polar amino acids. These are also called as hydrophobic amino acid because these are insoluble in water. The R group can be either have alkyl groups or aromatic groups. So the R group in non-polar may be either alkyl group or aromatic. Aromatic have benzene ring in the side chain. So first one is alkyl group, which is the non-polar amino acid. It consists of glycine, alanine, valine, methionine, leucine, isoleucine, and proline. So here the example of alanine is given, glycine and alanine is given. So you can see here, this is glycine and this is alanine. So you can see these contain alkyl groups. So this is the first type of non-polar amino acid. Next is the aromatic group. It consists of phenyl, alanine, and tryptophan, and we all discussed that it contains benzene ring as a side chain. So you can see here, this is tryptophan, and this is phenyl alanine. So these are aromatic groups, which is the types of non-polar amino acids. The next basic types of amino acid is polar amino acids. Here the side chains of amino acids contain polar groups like amine, alcohols, etc. and they are divided into three categories. The first one is acidic. Here the side chain has extra elements of carboxylic component and this have the ability to donate hydrogen atom. For example, aspartic acid and glutamic acid. These are acidic in nature. So you can see here, this is the aspartic acid. So this is one carboxylic group. This is another, so it has extra carboxylic group and it is acidic in nature, and they can donate their hydrogen atom when needed. The next type is basic amino acid. They have extra N group that attract a hydrogen, and they accept hydrogen atom because it is basic in nature. For example, histidine, lysine, and arginine. You can see lysine. So it have an extra amino group. So you can see, and it's another, and it behave as base and can accept hydrogen atom. The next type is neutral. They have neither acidic or basic and have an equal number of amino and carboxylic groups. So they are neutral. For example, serine, threonine, asparagine, glutamine, cysteine, and tyrosine, etc. So you can see here the acidic and basic. You can see these has the equal number. They are neither acidic nor basic. They have equal number of amino and carboxylic groups. So these are the neutral types of amino acid, which are the category of polar amino acid. So this is all about proteins introduction and structure, properties, and types of amino acid. Thank you for watching and joining.